So after the post-fertilization within the flower, we can see what happens to the different floral whorls. That is the fate of all the whorls. So let me draw a typical outline of a flower with the 4 floral whorls. This is the carpel, stamen, the male reproductive organ, petal, and the last whorl, sepal. Within the carpel, that is inside the ovary, the ovule is located, and we know the structure of the ovule. It is surrounded by integuments, 1 or 2 integuments, horseshoe shaped structure. So this represents the ovule, the white structure. Now what happens after post-fertilization? The different floral whorls, namely the stamen degenerates, the petals degenerates, and the upper part of the carpel, that is the stigma and style, also degenerates and falls. So what is left? Is the ovary with the ovule. Now, this is the ovule. After fertilization, the ovule becomes the seed. Now, this ovule is surrounded by integuments. The integuments becomes the seed coat. The ovary becomes the fruit, and the ovarian wall, that is the outline of the ovary, the ovarian wall becomes the fruit wall, that is the pericarp. So this is what that happens after post-fertilization. Which are the structures? Ovule becomes the seed, the integuments become the seed coat, the ovary becomes the fruit, and the ovarian wall becomes the pericarp. So now we know what is a seed. Seed is a fertilized ovule. What is a fruit? Fruit is a fertilized ovary. Now we are going to see different types of fruits. They are broadly classified into 3. True fruits, parthenocarpic fruits, and false fruits. Now true fruit, as earlier mentioned, ovary after fertilization becoming the fruit is called a true fruit. Ovary after fertilization becomes a fruit, then it is called true fruit. Whereas parthenocarpic fruit is just the opposite of true fruit. Always there's a chance of true opposite is false. So you have a tendency to write for true fruit, what is false fruit, you have a tendency to write it here. But actually the opposite of true fruit is parthenocarpic fruit. So ovary becomes the fruit without fertilization. Fertilization does not occur. So that's exactly opposite. True fruit, parthenocarpic fruit. Now false fruit. We know what becomes the fruit is the ovary. But in false fruit, other whorls of the flower. We know the other whorls, sepal, petal, stamen. So other whorls, when they contribute to flower to the fruit formation, it is known as false fruit. For example, in certain fruits, the thalamus contributes into the formation of the fruit. Not basically the ovary, the thalamus along with the ovary contribute to the formation of fruit. In that case, it is known as false fruit, and the examples for this are apple, cashew, and strawberry. So that's about the types of fruits. Next, types of seeds. Now, as earlier mentioned, what becomes the seed or what is destined to become the seed? The ovule. The ovule becomes the seed, and what is surrounding the ovule is the integument, and the integument becomes a seed coat. So that is going to be the true seed. And because seeds are the product of fertilization, we use the terminology hybrid seed. Hybrid seed or true seed or normal seed. And the other type is the apomictic seeds. So, two types of seeds, hybrid seeds and apomictic seeds. Now, as mentioned earlier, what is hybrid seed? Ovule after fertilization, just like the ovary. Here it is the ovule after fertilization becoming the seed is known as hybrid seed. Ovule becomes the seed after fertilization. So what will be apomictic seeds? Just the opposite of this. Ovule without fertilization occurring, becoming the seed is known as apomictic seed. Just like we have the fruits, true fruits and parthenocarpic fruit. In true fruit we have the ovary after fertilization becoming the true fruit, and ovary without fertilization becoming parthenocarpic fruit. Here there is exactly same, the hybrid seed and apomictic seed, ovule with fertilization and ovule without fertilization. But a little more in detail, you have to analyze this in detail. In both the cases, a bit more in detail. In both the cases there are embryo. See, the first case definitely we know, because embryo is a product of the fusion of male gamete and female gamete, definitely an embryo is formed. So there we know the presence of an embryo. So here we have an embryo. And we know the ploidy or chromosome number of the embryo is diploid. So I'm writing at 2n. How did we get this embryo? Because the male gamete fused with the female gamete. When the male gamete fuses with the female gamete, n plus n gives 2n. We have an embryo which is 2n. Okay, so in the first case, the ovule comprises of an embryo which is diploid. The second case also we are saying the ovule comprises an embryo again which is diploid. And at the same time we are saying no fertilization is taking place. So let's analyze the situation. Here male gamete fuses with the female gamete. So n plus n gives 2n. Then here it's only the egg, that is the female gamete, n, that is becoming the embryo. Now, how did n become? Just apply your 11th standard level knowledge. Just apply 11. When a cell is n, that is haploid, how will it become 2n? Answer simple as that. How did n become 2n? Or correctly saying is the gamete mother cell. Egg is formed from, we know by megasporogenesis, egg is formed by a gamete mother cell. The gamete mother cell is definitely 2n. And this gamete mother cell gives rise to the egg which is n. So how did 2n become n? First we must analyze how did 2n become n. Because of which process? Is it mitosis or meiosis? Answer, it is meiosis. Now I am proceeding from this stage. Now this egg is not going to fuse with a sperm or male gamete, and it is going to form an embryo. which is 2n. So how is n going to become 2n? The answer is here, how is n going to become 2n? There's no option for this. There's no cell division where n increases into 2n. So here, the correction should be made here. So I'm rewriting. This gamete mother cell did not undergo meiosis. I'm striking meiosis. It underwent mitosis. So what will we get? An egg, which is 2n. I'm rewriting. It is not haploid egg. It is a diploid egg. And this diploid egg simply develops into an embryo, which is 2n. I hope you have understood. Once again, the usual procedure of gamete formation is the gamete mother cell, which is always diploid, undergoes meiosis to form haploid gamete, and the haploid gamete fuses with the male gamete to form a diploid embryo. So this is the usual method. Gamete mother cell is always diploid. It undergoes meiosis to give the gamete. Here in this case we talk about the female gamete egg, and because it's meiosis it becomes n. And if this n becomes the embryo, how did it become the embryo? This egg fused with the sperm, male gamete. So n plus n gives 2n. So this is the case in the hybrid seed. What is depicted in yellow happens in the hybrid seed. Whereas in the apomictic seed, what has happened is, I'm rewriting from the normal and we are deviating. The gamete mother cell is 2n. What happens is, it gives rise to the egg, which should be n, but here the egg is 2n. So what may be the reason of this? Which division? Is it mitosis or meiosis? It is mitosis. So here we see the difference. Here it is meiosis in the first case, here it is mitosis. And this diploid egg does not fuse with the sperm. Here sperm is present, here no sperm. It directly develops into the embryo, which is 2n. So this is a very clear distinction between a hybrid seed. This is a hybrid seed, and this, what is depicting below, is an apomictic seed. In both the cases there are embryos, and the embryos are diploid. But how the diploidy? The first case, the diploidy is a normal method. The gamete mother cell, which is diploid, undergoes meiosis to form the haploid gamete. This haploid gamete, female gamete, that is the egg, fuses with the sperm, the male gamete. So definitely common sense, n plus n gives 2n, we have the embryo. But in the second case, we have the ovule with an embryo 2n. How is this? Here the deviation occurred here. There is gamete mother cell, which is diploid. Instead of undergoing meiosis, underwent mitosis. So it produced eggs which are diploid itself. And these eggs did not fuse with the sperm. They directly developed into the embryo, which is 2n. So this is hybrid. This is a case of hybrid seed, and this is the case how apomictic seeds are produced. And this process by which apomictic seeds are produced is known as apomixis. It is found in species like Asteraceae and grass. Now, in the field of agriculture, in the agricultural industry, hybrid seeds and apomictic seeds, which are preferred? Can you guess? It is the apomictic seeds. The apomictic seeds are preferred over the hybrid seeds. One reason is there's no segregation of characters. Why there's no segregation of characters? It's not the product of sexual reproduction. Here, egg from one parent and sperm from another parent fuse. So there is recombination, or there is segregation of characters. Here it's pure. The character of the egg is found in the embryo. So no segregation of characters. So we get pure lines. The second advantage in the field of agriculture is it need not be produced year after year. No need of these apomictic seeds to produce year after year. And last advantage, compared to the hybrid seeds, the apomictic seeds are cheap. So these are the reasons why apomictic seeds are preferred by farmers to hybrid seeds. That's why certain hybrid seeds are made into apomictic seeds. I hope you have understood this. Next topic is polyembryony. Polyembryony. As the name suggests, you can get a clue. What is polyembryony? Poly means many. So many embryos in an ovule is known as polyembryony. So here I'll have to draw the structure of an ovule. We know the structure of an ovule, horseshoe shaped structure, surrounded by integuments. See, look, now I have drawn one integument. You can split it and give like this, which means it is surrounded by two integuments. It varies from species to species. So you can either draw one integument or two integuments. Okay. Now this is the ovule. Now we know inside the ovule there's the embryo sac. There's the embryo sac, and definitely within the embryo sac you know about the 7 cells located, 8 nucleate, 7 cell stage we have already. That is, there are I'll just roughly give the distribution: antipodals, central cell, and the egg apparatus. So that is the embryo sac. Now all what we need to concentrate in polyembryony is just 3 regions. The 3 regions are integument, embryo sac, and the most important region between the embryo sac and the integument. Which is that region? The region between the embryo sac and the integument is the nucellus. I have highlighted in pink color. That is the nucellus. Now I am going to draw the nucellar cells. Homogeneously, compactly arranged cells. Nucellar cells. The nucellar cells. Now we are going to deal with polyembryony. What happens here? The nucellar cells divide and redivide. So these violet color, they divide and redivide, and they protrude where? Into the embryo sac. See? It's protruding into the embryo sac. And as a result, you see inside the embryo sac many projections. These projections are called embryo. Look at the number of projections in this diagram. 1, 2, 3, 4, 5, 6, 7. So we can see 7 protrusions into the embryo sac. How do these projections come into the embryo sac? The nucellar cells divide and redivide and projects into the embryo sac. So such an embryo sac with many embryos is known as polyembryony. Now here the question comes, what is the ploidy of these embryos? The ploidy of these embryos, it is 2n. The reason is that it is formed from the nucellar cells. The nucellar cells are 2n, diploid. And I told you they divide, redivide. The division here, it is not a gamete. Divide, redivide. Any division other than gamete, it is mitosis. So they divide and redivide to form projections, protrusions. By which division? By mitosis. And definitely we know mitosis, what will be the ploidy? It will be 2n. That's why the embryos in polyembryony is also 2n, just like apomixis. So in both apomixis and polyembryony, we have the embryos that are diploid, not haploid. And how did it become diploid? The reason is, even without fertilization, we know the reason. In the first case, how it happened, and the second case, how it happened. The main reason is because of the process of mitosis. And polyembryony is found in fruits like citrus and mango. So questions are indirectly asked. If you squeeze the citrus seeds or mango seeds, you can see a number of embryos projected out. What is this phenomenon? So the answer is polyembryony. The embryo sac consists of a number of embryos. And remember the ploidy of these embryos, it is diploid.